Good evening to all the participants. I think I have shared my screen. Um, yeah, I can able to see this biological databases. So can we recap the last session contents? Okay, so as usual, I mentioned that we'll be recapping the last session content so that we can keep in touch, and even I'll be more motivated from you. Like even I will be getting how much you understand. Yeah. So my first question is, what are the topics we discussed in the last session? So kindly type the answers in the chat box. If not possible with you, you can unmute and answer. What are the topics discussed in the last session? That is on yesterday. Yeah, just one day is completed. See, we have to move for 22 sessions. I don't know how you can keep hold of the concepts. Good. From Pooja, very good. So you have initiated, you mentioned about molecular sequences. That is, we discussed about various types of molecular sequences. Especially, we focused on, but okay, that's not mentioned. But difference between computational biology and bioinformatics. Very good. From Gia, Raiba, Gia. Introduction, we have discussed on biological databases. Good. And what else? Biological databases, difference between computational biology term and bioinformatics terms. And what else did we discuss? Did we discuss anything in detail? Even if not, I have taken hands-on experience. Whether I have taken any concepts in detail, molecular sequences alone. Okay, now let me just show you the slides and recap what exactly we discussed in the last session. Okay, good. Nice interaction. So we started with uh, the concepts like what is computational biology and what is bioinformatics. So bioinformatics, I already mentioned, this is a creation of tools that solve the problems. That's what we discussed, isn't it? You know how the bioinformatics term is coined? This is coined from Greek word. Okay, from the Greek language, we got this term called bioinformatics. Bio is generally we mean molecular biology. Info here we mean information technology. Okay, and matics is a statistics from mathematics. So what do you understand from this? Defaultly, if you see the textbooks, majorly in biology, they focus on molecular biology, information technology. So where you find large number of data present in organized databases. If you want to mine the data, like if you want to retrieve the data, or if you want to populate the data in the databases, okay, so of course you need even mathematics because you don't know how much data you have. So also you will be using the statistics, okay, which comes under mathematics. So totally this is a combination of three different subjects. Got it? So bioinformatics you find like this, and computational biology, the name itself implies that you're applying the computational part in the biology. That is, you're using the already existing developed tools from bioinformatics in the applications of various biology. So biology, here we can take all the biology, cell biology, molecular biology, immunology, okay, whatever you study, okay, so everything, okay, if you're using the tools and if you're studying the types of biology, then the application part, we call it as computational biology, okay. So again, we just recapped what exactly is the difference between bioinformatics and computational biology and molecular sequences. We discussed generally we have ACGT or the base pairs present in both DNA as well as RNA. For proteins, you have 20 amino acid letters. Okay. And if you want to see, if you want to convert DNA into protein or DNA into an RNA, obviously DNA to RNA is the same sequence. If you convert DNA to RNA, whatever sequence you find in the reDNA sequence, this string, okay, the same string will be appeared once again. Okay, but if you want to translate your protein, so you know that 61 codons, okay, code for 20 different amino acids, 20 essential amino acids. So the triplets of DNA sequence, or what we call it as a codons, code for a single letter amino acid. This is how we can convert DNA into a protein. So there are multiple programming languages like Perl, Java, Python, R programming. So in these programming, we have predefined packages. In the packages, we have predefined functions, especially to handle strings, especially to handle protein sequences, DNA sequences, protein structures. Okay, we have special functions. So instead of writing a lengthy code in C or C++ language, you can use these languages. In Java also, we have BioJava. BioJava is exclusively meant for working on biotechnology applications. So whatever I mentioned, reading the sequence, writing the sequence, that is more about the DNA and RNA protein. So we have predefined functions. The code is reduced as much as possible in these languages. Got it. Okay, so why I'm mentioning all this is because in after five classes, we'll be discussing about sequence alignment. In sequence alignment, we see blast tool, FASTA tool. These are used for pairwise alignment. Even these tools are developed. These tools are present in popular domain databases. So whatever tools you find there, okay, also these are developed only through these languages. Can be a Perl or Java. They have implemented from these languages because they have uh, predefined packages and functions inside. And if you see this biological sequence and structure databases, okay, I mentioned you about various biological databases, not limited to the sequence and structures. I've also gone for the other molecular sequences, like gone for the enzymes. Okay, what are the exclusive enzyme database? Okay, and we have seen sequence motif database, that is secondary databases. Okay, and we have seen pathways and complexes. So we just discussed about all this. Okay, so today I told you I'll be taking you to the hands-on experience on these two databases, isn't it? So especially from enzyme database, from Brenda, disease database like OMM, plenty of disease databases are there. Only one example they mentioned for each and every category of databases. We can see at least one or two. Okay, so whatever mentioned in this slide. So can I go to the web browser and can see each and every database in detail? Okay, so let me take you to the website. And participants, if you want to have connection with me, okay, so I'm just dropping my LinkedIn URL. Okay, if you're interested, you can connect with me. Okay, now let me take you to the website. So first we'll be seeing NCBI. So for going to the NCBI, either type like ncbi.nlm.nah.gov. Okay. Again, I'm telling you. So the URL for NCBI is ncbi.nlm.nah.gov. So NCBI is National Center for Biotechnology Information. Then what is NLM? National Institute of Labor and National Labor of Medicine and National Institute of Health and Government. Here we mean what government? What is GOV here? So not labor, this is library. National Library of Medicine, National Institute of Health, okay, and GOV. What is GOV? Whether is it, sorry, whether I close the WhatsApp. Whether it implies Indian government? What government is that? Good. USA government. Good, good. Superb. So here we mean .gov is a USA government. So it's a USA government website. Bioinformatics initially is a second large database when compared to DARPA. There is one history. There is one history behind the origin of internet. So if you see the history behind the origin of internet, okay, so the internet originally derived from a word called DARPANet, that is Defense Advanced Research Project Agency Network. Defense Advanced Research Project Agency Network. This is a US military. This is for US military services, which is a very largest. Okay, at even in 1970 itself, we find more than lakhs of people are there. Okay, so nearly some 1 million people are working in this US military agency. So during that time, they want computers, and they want computers connected to a network. So they initially have computers linked with LAN, and then also tried with MAN. But just because the computers population increased, so they try to have a kind of internet-based services. So just started with ARPANET. Later, they came up with a term that a person has renamed this ARPANET as Internet. So during the internet time, okay, even World Wide Web came, okay, and whatever websites you're going to type, okay, it is going to have a URL with the names, okay, like even the Google.com will be, you can find initially www World Wide Web, okay. So this is how the internet came into existence, okay. So at that time, we find large database from the US military agency, but nowadays, if you see the largest database, you can find only bioinformatics, because we have sequences, structures, so many. Okay, if you see the next generation sequencing, each and every patient, okay, if they are facing a rare disease, they have to go for genome sequencing. Like how if you go for consulting a clinician, if you have something like even SARS-CoV-2 infection, if you want to know how much infected, like 50% infected in lungs or 70% infected in lungs. Okay, if it is not curable for 1 week, okay, even the doctors recommend you to take X-ray photo, X-rays. Okay, so you have to take an X-ray, and you also have to take other tests also, RT-PCR, etc. Okay, so whatever you take, okay, so these are some of the essentials needed even for infections. But how about for cancer? How about for the uh, rare diseases or syndromes? Definitely have to sequence the genome. So each and every day, lakhs of genomes are sequenced in the world. So all these things are populated. Each and every genome is nearly gigabytes. Okay, you know the size of the genome. So that much gigabytes information is going to be stored. It's more than more GB. Okay, more GB is stored for in the even the hospital. So obviously, one should know how to handle this data and how to manage all this data. So only we are using this bioinformatics, okay, and linked with computational biology and try to find the solutions for it. So the largest database, if you see in bioinformatics or computational biology, we can come across this NCBI, which is maintained by National Institute of Health. Okay, so even if you find in 2019, when the COVID-19 infection was declared as pandemic, okay, at that time from epidemic to pandemic transition, you can able to find uh, no drugs or vaccines on the market. But if you see at that time 2019, okay, so more information are given about SARS-CoV-2 in this website. Okay, so they have more funding to work on uh, the upcoming diseases, infectious diseases, or even any other kind of a cancers. Okay, so they are the top leader in research to get the data. Okay, so which we find very difficult in our countries. Okay, now let me take you to the databases. If you see the databases, okay, even the father of NCBI is what we call it as father of bioinformatics. Okay, generally it's not a father, she is the mother. Okay, Margaret Dayhoff. So Margaret Dayhoff is one of the director of NCBI, who is also the discoverer of PAM, percentage accepted mutation, one of the amino acid substitution scoring matrix. Okay, so here it's Margaret Dayhoff is one of the director of NCBI who started this uh, database. And if you see the databases, okay, you can find projects information, some projects information on environmental samples, or if you if you want to work on projects, you can go with this. Okay, and the popular database what we use is CleanWare. Okay, the CleanWare. is purely a medical database, like the clinicians, okay, whatever they take from the patient, all the data of the patient are stored in the form of a data sets in CleanWare database. Okay, the MBBS MD persons will populate the data here. And if you see this gene, okay, this is the largest database for gene. Okay, what is gene and what is the difference between gene and nucleotide? See, here we have a nucleotide database and here we have a gene. Is there a difference? If so, what is the difference between gene and nucleotide? Can someone tell me what is the difference between gene and nucleotide? Because we have two different databases in NCBI. Difference between gene and, can someone answer for this? Right. And I think nearly 40, 35 to 40 are PG and PhD. So I think you have some knowledge on this, isn't it? That gene combination of nucleotides. Okay. Still, I can't understand combination of nucleotides. Nucleotides are the building blocks of genes, which are short pieces of DNA that carry. Okay. Arrangements of nucleotides make a gene. Oh, gene is the coding. It's a good. You got this. Okay. Generally, so I think Khan, okay, you have given some kind of a clue on this. Like, the genes are of two types of genes. Okay. The genes are classified into two types. One is coding gene, also called as exon. And what is non-coding gene? This is what we call it as intron. Okay, so the exon and introns are overlapped with each other, isn't it? Or merged with each other, not overlapped, or merged with each other. You have multiple exons and introns in what? In nucleotide. So you have a combination of exons and introns in a nucleotide. A nucleotide is a large sequence. Okay, in that, if you are focusing on particular coding regions, where if you match 2 or 3 exons, you may form a gene. Even a single exon can form a gene, or 2 or 3 exons alone. So then, how do you join the exons? You splice the introns. If you have exons, and I think I can show you, I can explain you through paint. I think you can understand, but still I'm giving you some kind of an example. So think these are introns. Okay. Now I'm going to draw the, okay. So I'm going to draw first initially introns. Okay. Now I'm going to draw the exons. If I try to splice, okay, so there is one process called splicing. If I do a splicing, okay, if I do a splicing, so this will be deleted, obviously. This will be deleted during the splicing. So obviously, what you'll be getting? You'll be getting only the matching of exons. Okay, so exon 1 and exon 2 are matched. And whatever you find here, this is what I call it as gene. This is what I call it as gene. Okay. So initially, whatever you have seen, the full sequence is a nucleotide. Okay, in a nucleotide, you have coding genes called as exons. A non-coding genes, the black color regions are introns during splicing, which is a molecular biology technique. Okay, the introns are removed and the exons are matched, which can form a gene. Okay, now we understand. So, do you understand now what is the difference between a gene and a nucleotide? Okay, now I think, okay, you understand even Meha and Akshita. Yes, good. She answered right. Okay, now let me take you to the thing. Then how you can observe and change if you want to try initially. Okay, so you know some fundamentals, but if you want to check what exactly you get as an output for a gene, try with one keyword. I suggest you to use insulin as the smallest protein, which you find less characters in the sequence. Instead of going with a large protein, If you try with the insulin, okay, it can be a gene, nucleotide, protein, all this, the length of the sequences will be less. So it will be easier for you to handle. Okay. So for our examples, okay, for our illustration, from the first class to the last class, I will be explaining you only with Homo sapiens. I try with only the Homo sapiens human thing, because one should know about the human information, okay, human disease information, human gene information. Then you can focus on even other animals if you're working on. If you are a veterinarian and if you want to work on that, you can try with the event dynamics. Okay, now I'm going to try with the gene insulin. So gene, so what do you infer from this? Now I told you it's a combination of exons, isn't it? But what is the ultimate thing in terms of molecular biology? The gene is a part of an mRNA, isn't it? It's a part of an mRNA, that too it's a kind of a coding mRNA. That's what you can understand from that. Okay, so generally in gene, you can find the mRNA kind of a sequences. The same, if you try the same, instead of gene, if you're trying with the nucleotide, okay, so the mRNA in the map to the chromosome, that information you can get from that. But instead of gene, I'm going to try with the nucleotide. And if I want to try with the Homo sapiens, see, you have already mRNA, you have DNA. Okay, so generally you find linear DNA, circular DNA as the types of DNA in the nucleotide. Also, you can observe linear mRNA in mRNA. Okay, so totally three kind of a sequences you can observe while you select nucleotide database. And initially I typed insulin, but later what I did, the right hand side, I used a filter called Homo sapiens. So this is a kind of an advanced filter. If you want to use an advanced filter, you can use logical and give a kind of a second thing. Okay, so you have to exactly give like Homo spacebar sapiens. Or else you just click that, obviously you get this kind of an advanced filter. Okay, it searches and it gives us plenty of information. How much information you got? For a insulin, human insulin, you got 11,519 entries. You have only one sequence for it, isn't it? But why we got this much entries? I told you, no? Even in Chennai, okay, one person is working on human insulin and he got a sequence in a sequencer. He can deposit the entire sequence, whatever he got, okay, nearly the 12,000. So he has deposited this. So you can able to deposit using the portals like Bankit, okay, Sequin and Bankit. So generally, these portals are generally used for depositing the gene sequences. Can be DNA, RNA, including a protein sequences. Okay, and whatever I open initially for you is a FASTA format. FASTA format. So this is the common format for all the molecular sequences. Okay, can be the major molecular sequences we handle in the bioinformatics projects or DNA, RNA. So just I opened what exactly I opened. I opened RNA, isn't it? So I opened RNA. So just go back and see. This is a linear DNA. Now even I can open the RNA and show it to you. If I open an RNA, still you can find only ACGT. mRNA is a small in length, isn't it? So you find a small sequence when compared to that. And that too, it's not a full mRNA, it's a partial, okay, complementary determining site. Complementary determining site, this is what CDS. So generally, if you see the FASTA format, okay, so what you can find is the pipe symbol. You know, this bioinformatics, the major of the bioinformatics applications before 20 years, okay, we use only Unix operating system, Unix or Linux operating system to handle these data. So all the databases can be NCBI, CAG, Uniprot, Swissprot, EMBL, all the databases, whatever developed by different countries, okay, they use even the servers, whatever they use to deposit the data, to retrieve the data, to mine the data, whatever they want. Okay, all these things they controlled only with the Linux server. You know, what is the reason behind why they have used the Linux? Because the Unix or Linux, is virus free. So these data are public domain data, public domain health data. Okay, or if you see this nucleotide, it's a databases, public health domain databases. So this should be publicly available free. Then only one can able to do research and can come up with a kind of a solutions for it. This is the main aim. Okay. But if you see in other engineering branches, for example, meteorology, materials technology, materials engineering, kind of the mechanical subpart, even the 3D structure of an alloy, if you want to download one structure of an alloy, you have to pay $10 to $100 based on the complexity of alloy. So most of the engineering, okay, downloading that 2D, 3D structures, okay, they have to pay for it. But for us, the entire data is present in NCBA is freely available just to have a common motive that anyone can do research, a genuine research, and can provide a thing. Okay, so that's the aim, of course. But you find multiple entries. You are as a researcher, you have to search. If you want to search for a full DNA, you have to search exactly where they are mentioned as full DNA. And you can take and download the DNA. So this is complete CTS. It's your duty to find the complete gene if you want to do, if you want to take the gene for further research. Okay, it's your duty to take even the mRNA, which is going to express the functional part of a insulin. Okay, you have to search and you have to search the journal papers, etc. Got it. Okay. So you can understand in nucleotide, you can find three types of sequences: linear DNA, circular DNA, and linear mRNA. Now let me take you to the next thing, genome. So in genome, okay, so generally you can give like a Homo sapiens, isn't it? So there is also so many so not only you have the animals genomes, you also have plant genomes. Okay, you have plant genomes, you have microbes, bacteria, fungi. So all the genomes are deposited in NCBI. So in NCBI, there is a database called the genome database called NCBI genome, where you can search the information of that. Okay, so now I take you to this. Okay, so can find a genome assembly, and you can find gene bank, etc. So I take you to the Homo sapiens. So it will take you to the genome reference consortium. If you go to the genome, it generally takes to the genome reference consortium. Okay, so it takes some time to download actually. Yeah. So if you see this coding genes and non-coding gene information, also not only find coding and non-coding, you can also find pseudogenes, can also find pseudogenes, small RNAs, etc. Okay, so to know more about the genome, while I explained during the 18th or 19th session, you can get more details from me. So now we can find some information of the human genome, how much nucleotide sequences, how much protein sequences, how many 3D structures are available in NCBA. Okay. Gene expression omnibus data sets are available here. We want to go to check the expressions of a particular protein, you can find from here. Okay, so this is something how a human genome is linked. Even if you want to blast an entire genome, we have an entire genome here in NCBA genome. Okay, so you can check each and every chromosome and the genes present in the chromosome in the human NCBA genome. Okay, now let me take you to the next because this is a vast thing. Okay, like it's a version where we can see so much data inside if you click hyperlinks subsequently. So now let me take you to the next database. I told you even the genome is linked with the GEO database, sets, data profiles, etc. And now we can go with the ONIM. Okay, so this is actually online Mendelian inheritance in man. So one should know actually, like even before going to the OMM, one should know the various database identifiers. Like if you want to download the complete set of the human insulin gene, a linear DNA sequence. So if you know this accession number, this is what we call it as GenBank accession number. This we call it as a GenBank accession number. This is a 8 characters in length. This is an alphanumeric code, okay, which is 8 characters in length. Then what is this point to? This is the version. Okay, I think last 2 years, I think we got this kind of a version. Earlier we have only point 1. Okay, now also we got point 2 version. Okay, and after 10 years, we may get a point 3 version. Okay, so this is one of the database benefits, like how I can identify a student through roll number, registration number, name, etc., including initials. Here, the data of human insulin, linear DNA can be retrieved through this. Either you can type the same here to find the exact information, only one entry you'll be getting now if you copy and paste it, or else if you don't know anything, you are a layman, you are a new researcher, you want to work something on this, then you can type the keywords. Start with a protein and then check with a species for screening. And this is the second one, second database identifier, which we call it as Gen Info ID. The Gen Info ID is 10 characters in length. If you see 15 years before, 10 years before itself, we can find only 8 characters in length. But now you can find even in 10 characters in length. See, this is varying 6 characters in length. Here now you get 10 characters in length. Okay, so the length is increased up to the 10 characters. So even if you see some 20 years before, you might have seen this 6 characters in length. Okay, you got it. Based on the population data, even the Gen Info ID length is can find the variation of. And now we can just link all these concepts in the PPT. So I told you how to go for searching a nucleotide, how to search for a gene, okay, and we have seen even how to search for a genome and how to link with the GEO, okay. So then what else we have to see? We have to see how to download the chemical structures, okay, how to download the chemical sequence, okay, and what are the various formats of the nucleotide and protein sequences. Okay, this we'll be seeing it now. So just we started with gene sequence and nucleotide sequence, etc. So what are the various formats? I told you one common format which we find in almost all the databases can be NCBI, NCBI. There is another website called Uniprot, uniprot.org. So this is a database. Okay, this is a database maintained by. So here in Uniprot, default it takes to the insulin keyword. Okay, so you have only one entry for human. But why we have few entries for human is here you can find the protein name is different. This is insulin receptor. This is insulin degrading enzyme. Okay, there is also insulin ligand. For this, you have a different thing. So for each and every protein, you have only one entry. Unlike NCBI multiple entries, you have only one entry here. Okay, and the entry is a common 6 alphanumeric letters. Initially, the first letter is an alphabet, and the remaining 5 are the digits. So, okay, so each and every protein of a particular species have a unique Uniprot entry. Okay, so this is the URL is uniprot.org, which is maintained by UK government. Okay, the server is present in London. And if you see this Uniprot website, here also you find so many databases. Okay, so more about this we see after discussing from NCBI. After discussion about all the databases in NCBI, then we can go to the Uniprot and we discuss and we compare the databases present in NCBI. Okay. Now we can first complete this NCBI. Now in OMIM, if I want to search for a disease, for example, arthritis, I'm just typing arthritis, a disease information. You're getting nearly 486 entries. There are types of arthritis, nearly 100 types of arthritis are there. But you found only 486 entries. But each and every entry might be a different kind of arthritis. Yeah, this arthritis is different, this arthritis name is different. Okay. You find so many information on that. So in OMM, you can find a OMIM ID, that is for leukoencephalopathy. Okay, which we call it as also called as colitis, also called as hypogammaglobulinemia. So if you see in the Apollo hospital or in any hospital, they give some kind of a new name which we don't know anything about it. Then if you want to get some information, more information, either you can search in Wikipedia. If you are a biotechnology researcher, also you can search here. Okay, so you get more information on that. You get gene summaries, you can get genetic tests, you can get more medical literatures from here. Okay, so there are hyperlinks. This is a database where you can find, you can link with more data through the hyperlinks. Okay, so for example, if I click genetic test, okay, we can see what are the various genetic tests. The major genetic test we use is the fluorescent in situ hybridization. Okay, fluorescent in situ hybridization is generally used for karyotyping of chromosomes. Karyotyping of chromosomes, we use this technique, but it's an expensive technique. Okay, so using this, the arthritis, the type of arthritis, colitis can be characterized. Okay, so which is a heritable disease. Okay, which is heritable from fathers, that is from ancestors. Okay, if you want to find a solution, if you want to get a gene names, you can get the gene summaries from here. So what are the various genes responsible? So here there is no such information available, but for popular arthritis like rheumatoid arthritis, you get more information on even the gene summaries. If I click the gene summaries, now I can find so many genes are coming. Okay, might the Indian person, a known Indian person is suffering with this CHIF gene disorder, and a USA person might be suffering from a different gene. We don't know about that, but one has to check all this thing information from the OMM database. Now let me take you to the this is medical subject heading. Okay, like if you are focusing on a particular disease and you want to get some information other than heritable disorders, this is a common thing. Okay, if you want to find information of a common disease, you can search here. Okay, medical subject heading is what MESH, abbreviated as MESH. Now we can, if you want to download a protein information, click protein, and instead of a disease, type any protein name. Okay, type any protein name. How I type like insulin. So before typing insulin, I showed you even in Uniprot that in insulin you have insulin receptor, insulin ligand. Insulin ligand is generally going to bind with the insulin receptor to activate the signaling. Okay, then only the insulin function. Now obviously it regulates the glucose level in the blood. Okay, so if you want to find the insulin information for Homo sapiens, you can have nearly 7,083 entries. Whereas there in Uniprot, we find only 3 entries. Ligand receptor, and another is another kind of thing, a degrading enzyme. Okay, we have very few entries, but we have multiple entries, multiple repetitive entries. Okay, so but one has to properly select this. So even yesterday, one of the participants asked me, like, sir, we find so many isoforms, how to start with a project? I'm confused, terribly confused, how to go across. So I suggest you take only normal protein. Don't go for the isoforms. Okay, even you can see the isoform size is larger when compared to the normal insulin. Okay, so during the sequencing or even during the crystallization formation, where if your objective, if your problem statement is to get a 3D structure of a protein through any gold standard techniques, then obviously you have to prepare a protein, isn't it? So during the preparation, I get like this. But whatever they mention, okay, here, see, there is no such isoform mentioned. So this is the exact length of a insulin. Okay, this might be for an insulin receptor or something like that, but you can choose this. So only you have to use it. Don't even use this. See, the third one is also, they mentioned like insulin, but the size is less. Whether the size can be less, whatever size they mentioned as the maximum is the correct length of the protein. So even if it confuses with the multiple entries, don't go through this. If you have more confusions, I suggest you to go only with Uniprot. Go with the Uniprot. See here, the Uniprot, okay, for I told you in insulin, you have an insulin receptor, insulin ligand. See the size, what is the length of the insulin ligand? It is 110. So the Uniprot confirms you the total size of the protein. And if it is a receptor, you can find it. Might be whatever isoforms they mentioned is a part of the insulin receptor. Got it. So you just keep it and you just go through exactly, check with Uniprot and come up with a conclusion, and you can even download from here or you can download from there itself. But you know, if there are any engineer, if insulin is engineered, okay, you can't find here. You find information from here, but the sequence won't be present. But here you may find a sequence. In a native protein, in a wild protein, not only get a sequence for a wild protein, you may also get a mutated proteins, engineered proteins. Okay, so one should keep in, one should compare this using the pairwise alignment. And even if something is engineered, okay, you can check with it, and you can also try whether the protein is more stable when compared to the normal wild protein. Okay, there are plenty of ways and application areas even to check like this. Okay. Okay. Now I was explaining about NCBI. Let me complete about the NCBI. So one thing I showed you is the insulin, Homo sapiens insulin. Here also we have formats like FASTA. We also have a format called GenPet. See, generally we call this as a GenBank, but for a protein, we if you see this, for a protein, when I clicked the GenPep, you got like GenBank accession number. Even though it is a protein, even though database is a GenPep, but you get only the identifier, a database identifier, a GenBank accession identifier. Okay, so this is an accession number, GenBank accession number. This is I told you already, this is 8 characters, and point 1 is the version number. Okay, so whatever I opened is the GenPep format. Okay, here when compared to the FASTA, okay, here you find partial annotation information. First, I can show you the FASTA, then I can show you about how the GenPep format looks like. See the FASTA, what exactly you can find the information for an insulin protein. In a 110 amino acid, okay, the first 60 characters are present in the first line, and the remaining characters are present in the second line. Like each and every 60 characters will be trapped to the next lines, to the new lines. But what we have to understand, while if you want to download the FASTA format, one should remember that the first line is an identifier of a protein. The identifier here after greater symbol, you can find a GenBank accession number. And this is a name of a protein, which we call it as locus name. This is locus name 1, and this is locus name 2, which is the species name. Okay. So what are the various database identifiers for a nucleotide or a protein? One is GenBank accession number, and if there is a GenInf ID, you may also find GenInf ID. Here we don't have, okay, but you have the locus names. The locus names like insulin and Homo sapiens are the locus names for it. So this should be in one line, and whatever sequences you paste, this should be in second line. This is not a break. You should not assume that this is coming in the third line. Just because the viewers has to see properly, it should be a user friendly, like a graphical user interface. That should be a kind of an interaction. The viewing point, if we have to feel like pleasant, So they are breaking, they are wrapping in the next characters in the second line. So if you want to save this sequence in FASTA format, I suggest you to use the best editor as Notepad. Use only Notepad, okay, for the sequences. We have editors like Notepad, WordPad, Microsoft Word. Microsoft Word is very rarely used. Either you have to use Notepad or WordPad. For a sequence, better use Notepad. Okay, can be nucleotide or protein sequence. Now, I told you they should be in first line, isn't it? How about this? So this should be in second line. So I click there and click delete. So obviously you find entire sequence in the second line, but still some kind of break is there, some wrapping is there. You can check any word wrap is there. You remove that word wrap. Now you see all the characters of the amino acid sequence are present in second line. So this is the proper way of saving the FASTA format. Okay. So if you want to save the FASTA format, okay, somewhere in the desktop, okay, I'm going to give so default in Notepad save in text documents file type. Okay, keep it there. Don't change it. Keep it in text documents. But whatever you're going to save, like if it is a sample.fasta or insulin.fasta, okay, type the file name .fasta in double quotes. The file name dot the extension should be in double quotes. Okay, so this is how I can say. There are so many editors. We have even BioEdit for and other softwares for editing the sequences. Even I generally use Discovery Studio, okay, for saving the FASTA formats and to edit all this. But okay, even the Notepad, I think even if you don't have any bioinformatics softwares at present, you can try the same with the Notepad. Okay, just save it, file name .fasta in double quotes under .txt doc. Yeah, even if you're saving it in WordPad, save it in .txt documents. If it is in some other format like RTF, rich text format, definitely you get in trouble if you try to use that file as an uploading for any tool to work. Okay, so try with only text documents and save it as filename.fasta in double quotes. Or else what is going to happen? Filename.fasta.txt is your file extension. .txt is the original extension. Filename.fasta is your file name. Okay, it should not be like that. So try to save exactly how I mentioned. So there is a FASTA format. Simply what you go infer from this, what is the amino acid sequence of a protein is shown here. But so it is a no annotation format. No annotation. You don't find much information. Almost no or nearly annotation is found. But if you want to find some information, not more information, some information like taxonomy, like exactly we have searched for a human kind of insulin. The human is coming under taxonomy eukaryotes. Okay, under eukaryotes, you find metazoa classification, core data. So you can find the taxonomy classification here. And who is the first person to post this protein? Okay, who is the person who take responsible of posting the protein in the NCB? These are the authors. These three authors have taken that active role. They got this kind of an accession number. Okay, so when they have posted on 10th June 2016. Anyone can do this. Now, if you're doing some kind of an experiment, you can just send the sequence to any company. They give you a sequence, and if you find something new, or even if not new, simply post it. It's a free to post any sequences. Okay, so and if you see even what journal they have used for publishing this kind of a sequence, this is the journal name and more information and PubMed ID of even the journal paper is mentioned and some more information, and later the last is about the amino acid sequence. But this format generally you can't able to use it for other databases. So you can use only for the NCBI, you can't use it for the other databases. So generally the FASTA is the format, is the universal format used to take the information of a particular molecular sequence, can be a protein or nucleotide or a gene. Now let me explain about the next database. The next database what I'm going to take you is the illustration for PubChem compound. There is a database called PubChem compound. In the PubChem compound, you have to type a chemical name. Okay, it can be even a plant compound also. Okay, so for example, if you want to know more about curcumin, okay, you want to know some information like a 1D information, 2D, 3D structure information of the curcumin. You want to know about the physical and chemical properties of the curcumin. You want to know about the FTR spectra, okay, even NMR spectra. Okay, all the details are available in this database. PubChem compound, more detailed information. Okay, so even the PubChem is giving one kind of an identifier called PubChem ID for each and every chemical. Even if it is a derivative of a curcumin, like a curcumin glucuronide, okay, for this you have a unique kind of an ID. Okay, each and every chemical here. So you can see the chemical ID for this. This is the PubChem ID for this. Okay. So likewise, if you know the PubChem ID, you directly type or you can search like. So under curcumin, you find 129 entries. This is not repetitive, like how you find in GenBank and the for a nucleotide and protein. Each and every entry is a derivative. It's a different derivative. This is a curcumin. This is a curcumin glucuronide. This is the dimethoxycurcumin, diacetyl curcumin. Okay, likewise, whatever compounds you have, okay, in the curcumin. Okay, so you have a different entries for it. Now let me take you to the first entry, curcumin. So if I take you to the curcumin, okay, so what are the information you can able to find? PubChem ID, structure information of 2D and 3D. So these are just the images. If you want to download exactly the 2D or 3D structure images, get down. You can find that. Before that, you can see what other information you can find in the PubChem compound. So what exactly can able to find? You can find the description. You can find the applications, biological applications, where exactly we can use, whether it can use as an antimicrobial, especially it can be an antibacterial or antifungal, even whether it's acting like an, it's showing an anti-cancer activity, like showing an anti-neoplastic agent. Yes, it's also an anti-neoplastic agent. It's a hepatoprotective agent. Not able to show. No. But okay, anti-hepatoprotective agent, favoring agent. Okay. So, so many biological applications are there. And if you want to download the 2D structure, better click this download coordinates. Here you can find STF format. The first format what they have shown is the STF, structure data format. Click save. Okay, so if you click this, okay, some problem happened. What happened? I can't able to click. The system got hacked. Am I audible to you? Yes, I think now it's working.
Yes, sir, you're audible.
Good, thank you. Now I try to save it in the desktop. Okay, so what I'm going to do is I'm going to save the 2D structure of a curcumin. Okay, now I'm going to save this. Similarly, I'm going to save the 3D structure. Now check what is the difference between the 2D. So 2D structure looks like 3D structure looks like, which is going to have X, Y, Z coordinates. Here you have only X, Y coordinates. Okay, and if I like to download the same, I suggest you to use to download .stf format. There are also other formats, but the STF is a common format which is used in most of the visualizers to visualize the molecule. Okay, what happened? Again, it's taking some time. I'm saving again, once again, the 3D structure in the desktop. Uh, yeah, now I'm going to save the same in desktop. So whatever I saved in desktop, if you want to open it and visualize it, okay, we also have an visualizer tools here, okay, inbuilt in PubChem to see the 3D structure. But if I want to visualize using any visualizer, come on, system is too slow on today. Let me, so I found, mention it. Yeah, so I just take it to the desktop and open the 2D structure. I'm right clicking the file 2D structure what I downloaded. I'll simply double click. Okay, today my system is too slow. I don't know. My then I can take you on tomorrow, like how to see the sequences, how to edit the sequences, how to change anything in the sequences. All these things I'll be teaching you on tomorrow. Today I can complete the databases. I didn't experience such a problem. This is the first time I'm facing. Too slow. Mm. Okay. Hello.
Yes, sir, you're audible. We can hear you properly. But now your presentation is not visible, sir. I think sir left the meeting, na?
Am I audible now? Hello. Am I audible now? Yes. Sorry. How much time I can't able to connect? May you know how much time? 2 minutes got wasted. 2 or 3 minutes. How many minutes? I don't know. Can someone respond? How much time?
Yes, so you were audible till just a little while ago
Oh, oh, oh,
when you
oh.
started facing the technical issues.
Oh,
But that was
disconnected.
all.
Okay, okay, got it.
Yes.
Disconnected for 2 to 3 minutes. Okay, I understand. Okay, I don't know. During downloading, I think I got some trouble. But in the next session, I teach you how to open that 2D or 3D structured molecules. Okay, and also I teach you how to edit the characters, edit the sequences, can be protein sequences, etc. in the next class. Okay. Today, I just teach you what are the various databases present in NCBI and Uniprot. Okay. So the 2D and 3D structure generally I told you to download only the HDF format. The 2D you find one XY coordinates like this, and 3D coordinates you find like this. Okay. I think the screen is shared and it's visible to you, isn't it? Yeah. Yes. 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 Okay, now if you want to download the sequence format, better go for canonical smiles. There are two types of smiles format. One is canonical smiles and other is isomeric. I suggest you to use only the canonical smiles. Whatever you got a 3D structure like here, okay, this 3D structure information, this 3D structure information, uh, this 3D structure information, you can able to find in this single D format. This is 1D coordinate. This is 1D. We also have 2D and 3D to download. So to download this, okay, just copy it and paste it in Notepad or wherever editors you want to use it. Molecular formula and the CAS, the common identifiers. Even you can find some FTR spectra. You can find so much information, chemical, physical, chemical vendors. If you want to buy that, okay, you want to find who is providing you an economically better, okay, all this information food additives information, pharmacology, safety hazard, toxicity, literature, patterns. So all this information you can download from the NCBI PubChem. Now let me take you to the next database. After PubChem compound, okay, we can go for single nucleotide polymorphism information for a disease. Okay, if you want to find for a specific disease of SNPs, you can check like this. So you can give a disease name, or even if you know the target, go to the target, type the target protein name, and you can find that. And if you want to download the 3D structure of a protein molecule, for downloading the 3D structure of a chemical molecule, you are using the PubChem compound. But if you want to download a 3D structure of a protein, use the structure database. The structure database generally takes you to the what database? Yesterday I told you there is one structure database in NCBI. Can someone tell me what is the NCBI protein structure database present in? It's not PDB, but try it well. The PDB is a common database. Actually, for a PDB, you have to type like if you in Google, if you type like PDB, it defaultly takes to the rcsp.org. Okay, or even it can take you to the PDB, www.pdb.org. Either you can type pdb.org or you can type like rcsp.org. So this is the PDB database. This is not from NCBI. Then can someone tell me what is the exact database? No, not Uniprot, not FASTA. I think you have missed. The exact answer is molecular modeling database. You remember MMTB. So the structure database present in NCBI, that is here the structure is only for proteins, not for any other DNA, RNA, or can be chemical compounds. You can I just type insulin, you find nearly 1582 3 structure of proteins. Not just the human insulin. Here you can find the human insulin mutant. Here you also find some kind of insulin complex with some analogs, some hormone growth factor, insulin of Sususcrofa, not the human. Okay, if you want to download the human, you can also select advanced filter. Here also you can find what are the gold standard techniques used to get a 3D structure. One is NMR, nuclear magnetic resonance. The other is the X-ray crystallography structure. Okay, there are so many filters which you can use. Even protein DNA complexes, protein RNA complexes are there. So this is MMTB, and MMTB ID is there for each and every protein entry. And the common PDB ID is 1T1Q. MMTB ID is different, PDB ID is different. 1T1Q, if you type it in here, okay, obviously get the same protein. Not only in PDB and also in other small protein databases, if you type the PDB ID, it can take you to that. For example, PDB sum. There is one database called PDB sum. Even if you try this, you get a kind of structured information of that. Okay, this is something about structure. And like structure taxonomy, you have, okay, you have for bioassays. If you want to get some kind of a bioassay information, you can use it. Now let me take you to the Uniprot. At least we have seen few databases in NCBI. In Uniprot, majorly we go for universal protein knowledge base. So if you're a layman, if you don't know anything about your protein, but you want to work extensively on your protein in MS, MTech or PhD research, use this Uniprot. This is a fully annotation database. And unlike the GenPept or GenPept, which is a partial annotation and a FASTA, no nil annotation database, you better go for this kind of a Uniprot where you find much information on that. Okay, if I type even in about insulin, so what disease it is associated with and what cellular regions. So one can try all this. I can't able to narrate each and everything, but majorly from my side, from my research, I generally take not only the FASTA format, I take the protein-protein interaction information. If you're working on proteins, okay, protein bioinformatics, one should have a knowledge even on the protein-protein interactions. Okay, so which is most important even for disease related. If you are working on a cancer, so this I majorly use, and this also majorly is the structures. Here in structures, you have not only a single structures, you have plenty of structures are provided. Okay, so you can see I can minimize the font and show you. So you can see even a part of a protein. You don't find the entire protein. Even a part of a proteins are published in the form of a structures. Even the part of a structure is sufficient for your research. So not the entire 110 amino acid is giving the insulin function. Only a part might be some 13 to 15 amino acids, which is considered as a motif of a protein. A functional part of a protein, we call it as a motif, which might be a 13 or 15 amino acid of 110 total length of insulin, which gives the function. Okay. So even that part of a protein also can be published as a 3D structure. We can find a PDB-ID. If you are working on a therapeutic thing, research, then you can also download the thing and you can work on it. Okay. So insulin is a small protein, but if you are working on a very large proteins, and if there is a need to reduce the time for research for docking and simulation, better go for that even that part of a protein to work. Okay. Likewise, family and domains, sequence, isoforms. So one can try everything. Okay. Even the freshers, especially I found many UG students have joined. Okay. So you have to try today about for a keyword like an insulin and try with each and every database. UniRef, UniReference can give you a reference of proteins linked with a particular protein. If, for example, if you're typing the human insulin ligand, so what are the different proteins associated with the human insulin ligand? So that information, protein clusters can be found from UniRef. This is uniprotein archive. Only the FASTA format of a specific protein is shown here. So even you can find the sequence. Okay, the Unipark is available here. This is what Unipark, we call it as Unipark. Okay, if you want to download the 3D structure, I mean the FASTA format, you click here. Okay, directly take you to the Unipark database. Okay, and sequence clusters in Unitype. So this is the FASTA format you can find here. A small thing is shown here. Like you also have an exclusive proteome database, which we can't find in NCBI. We have a proteome database in Uniprot. Okay, and we have the informations. So one can try this. Okay, even if you want to download the 3D structure, you can type the 4-letter PDB ID, and it can so you can type any thing whatever you're working. Okay, try with it. So get a 3D structure. So to know more about this, we'll be saying during the protein structure determination class. Okay, today I have completed about the biological introduction of biological databases and how to practice on the databases in NCBI as well as Uniprot. Okay, now any questions you can ask me. Sir, can you tell me the names of software required for basic bioinformatics? Actually, at present, okay, I just am demonstrating only the databases. In the next class, okay, the Pyrex is a computational application. You know, the Pyrex can be, so during the project discussion, if you see here, okay, mini project discussion, okay, so during that time I can teach you. Okay, now I can't because we have so many concepts to learn. If you want to learn about the tools, okay, even in the next class I can teach you 1 or 2 tools, but majorly the tools so you can find from here. These are the tools. Okay, you generally can find here. Even these are the databases. How the data can be found, how can be edited, all these things you can see in the next class. Okay, and also what are the special databases present other than protein nucleotide? We'll be seeing the other classes. So tools, the practice of tools will be coming in between. Here also you can find tools. Okay, so tools and databases come in random. Okay, so you can learn from each and every class. Okay, sir, I was unable to save the SD format in Notepad. So this can we see it on tomorrow, Akshita? Definitely, I can take this. Okay, in the next class. And in other, I want to download insulin. Yes, you can download insulin. You can download it, but I suggest you to better go for downloading this BioVia Discovery Studio. Okay, so just type like BioVia Discovery Studio. Everyone can try this. Okay, try to download this. It needs a large data, of course. Okay, one can try with Wi-Fi if you have more space. Okay, so download this and install it. If you install this BioVia Discovery Studio Visualizer, tomorrow I can teach you how to open the insulin structures and how to edit all this. Okay, so presently you can download that BioVia Discovery Studio. And any other doubts? Today, you can try this as a task. Try with the NCBI and Uniprot and try with the Bioinformatics. GEO database. Yes, you have to just get the information. You have to type the disease name in the GEO, NCBI GEO, and get the information of the GEO. So if you're working on GEO, you can come to know more about it. So if you, okay, any other doubts? So for Anand Kumar, I suggest you to even go through YouTube videos if you're working on GEO. Okay, if you have interest on that. So what are smiles and the difference between canonical smiles? So tomorrow we'll be discussing on this. Okay, canonical smiles and isomeric smiles. Smiles, actually these are two types of smiles format. One is showing an isoforms, even the isoforms are shown or reflected. You know, D form, L form. Okay, the canonical is a common thing. Okay, you don't confuse between the D and L forms of compounds.
Hello,
So generally we use the common format canonical smile.
hello, sir.
Tell me, sir.
Sir, I want to download some
Yeah, you're right.
100 organisms, some specific protein sequence like DNA sequence. Is it possible to do in a single time, like in a single click? Is it possible to download from lower organism to higher organism?
Actually, we can go for going for blast. We can do the blast. Okay, we try the same in Uniprot. It's possible. It's possible to take all the sequences and compare at the same time. Okay, that I will be teaching you during the 7th or 8th session. So it is there in our syllabus. So I'll be teaching you. But in the second class, you can't expect everything. Okay.
Okay, sir. Sir, one more thing I have.
Mhm.
One doubt I also have. Sir, can I download like the information of some metal oxide nanoparticle is there in PubChem?
No, 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 no. That's not possible. You can't try it.
Okay.
Yes, you can't try.
Okay, thank you so much, sir.
Okay, participants. So I found many participants have many doubts, and you have so much interest to work on this. So don't leave your enthusiasm. Okay, try to find. Okay, try to explore. If you get a new phone, how you have an enthusiasm to learn something. Similarly, you can try all this. Nothing is going to happen. Your system is not going to get disrupted. Okay, so try everything. And if you have any kind of genuine doubts, I can clarify in the next class. Thank you. Have a great day to you all.
Thank you so much, sir.